ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.
Hello students, let us continue with chlorophyll. We are in the discussion about the structure of chlorophyll. So we understood very clearly the phytol head and phytol tail we just have studied, head part and tail part. Head part having 35 carbons, tail part having 20 carbons. Now let us continue. When you look at carefully here, let us make differentiate between chlorophyll A with a chlorophyll B. So let us make differences between chlorophyll A with chlorophyll B. Why we are comparing chlorophyll A and B here? Because we studied these two are nothing but these two are two main pigments of photosynthesis. Two main pigments of photosynthesis. These two pigments plays very important role in photosynthesis. That's why we are discussing what chlorophyll A and then chlorophyll B. Right. Now let us make the differences here. Now if you take chlorophyll A, this is nothing but bluish green. The color with what it appears is bluish green, whereas chlorophyll B is olive green. Or you take yellow green. Right. So chlorophyll A appears bluish green, chlorophyll B appears olive green or yellow green. Then where are the differences? Now if you take the molecular, what you can say, a molecular formula, meaning the structure contains C55, H72, O5 and 4. This is nothing but molecular formula for chlorophyll A. With what it is connected? Magnesium, of course. It's been connected with magnesium. Right. Next, coming to this part, then what is a molecule? And this is approximately, if you take the molecular weight, it is 893 Daltons. Molecular weight is 893. Coming to chlorophyll B, if you take the molecular formula, so what is the molecular formula which we get it here? C56, H72, O5 and N4 being connected to magnesium. Sometimes what happens, the two hydrogens will be less. Sometimes 72 will be there, that's also correct, and sometimes 70 will be there, that's also correct. So mostly this is the thing which you have to take. Okay, so here then where is the difference here? How difference will occur in terms of molecular formula? O6 also will come there, O6. Oxygen will be extra. I'll tell you why oxygen is extra and why variation in the count of hydrogen. I'll let you know, because the basic difference what you find here. In chlorophyll A, 4-pyrene head contains how many pyrrole rings? 4 pyrrole rings. A second pyrrole ring. Guys, try to remember, second pyrrole ring, third carbon. Second pyrrole ring having third carbon attached to methyl group. Then it is identified it as a chlorophyll A. Now coming to chlorophyll B, if you take, same I am writing 4-pyrene head, no doubt second pyrrole ring. In second pyrrole ring which place? Third carbon will be having a aldehyde group. So this is the group you find. What is the group here? What is the group you find here? This group is a methyl group. This group is a methyl group and this group is aldehyde group. This group is aldehyde group. See, that is the main difference. I repeat, So, porphyrin head, in that 4 pyrrole rings are there. In that second pyrrole ring, third carbon in the second pyrrole ring having the third carbon contains what? If it is a methyl group, then we call this chlorophyll A. If the porphyrin head second pyrrole ring third carbon is connected to CHO, that means aldehyde group, then that is chlorophyll B. So, these two are main differences between chlorophyll A and chlorophyll B, right. Now other you find what? Chlorophyll B, C, D, E are found in case of red algae, brown algae as well as found in case of blue green algae. So, they are nothing but what? Chlorophyll. Now, chlorophyll is also associated with other pigments. What are the other pigments which are present along with the chlorophyll? What are the other pigments which are present along with the chlorophyll? Are called carotenoids. Along with the chlorophyll, carotenoids also plays an important role in photosynthesis. Now, these carotenoids are of two types. In case of higher plants, what are they? Let us take as carotenes, try to remember, and other one as xanthophylls. What we can take? Carotenes and xanthophylls. So, when we take the carotenes and other one as xanthophylls, now if you take the carotenes, now if you take generally, if you take chlorophyll, if you take generally, this is a fat soluble and water insoluble. It fat soluble and it is generally, if you take chlorophyll soluble in fats and it is insoluble in water. Coming to carotenoids, generally carotenoids are lipid components. They are nothing but lipid components. So, when you take this lipids components, carotenoids are nothing but compounds of lipids, try to remember. Now, when you take the carotenes, now in this case if you see, carotenes can be differentiated with the molecular formula. I am writing molecular formula here, C40H56 are generally called as what? Carotenes. Coming to xanthophylls molecular formula, we can take C40H56 and oxygen. Very, very important. What is the difference? Oxygen. What is the difference? Oxygen. Similarly, I think I told you, when you take chlorophyll A and chlorophyll B, right. What is the molecular weight for chlorophyll A? Molecular weight for chlorophyll A 893 and molecular weight for chlorophyll B will be 709. I think I have not written chlorophyll B. Okay, this is 709. Chlorophyll A is having 893 or 863, from 863 to 893. Okay. So, here when we take the carotenes and when we take the xanthophylls, carotenes generally they will be yellow, yellow orangish color, or they will be yellowish red in color. Coming to xanthophylls, mostly they express yellow, mango, ripened mango, yellow in color, it is because of xanthophyle, yellow mango. Ripened mango, that is yellow in color, it is because of what? Yellow in color, right. Now, apart from this, thank you so much sir. So, this is yellow orange, this is yellow red in color, this is yellow in color. Now, apart from this, in case of xanthophyle, you find very important pigment called lutein. What is the important pigment? Lutein. Lutein is a most distributed yellow xanthophyll occur in foliage leaves, especially in autumn season. What's the meaning of this? Now when you take especially xanthophyll, most commonly found xanthophyll pigment is called lutein. So what is there to remember about lutein? Very important. Lutein is referred as most distributed yellow xanthophyll. You know, in autumn season, generally spring season leaves will come, new leaves, new flowers will come, right? Spring season that, before that autumn we find. At the end of autumn, what you can see, all leaves will fall off, or whatever the leaves are there, they will turn into yellow color. Yellow color of the leaves, especially foliage leaves, if you take, in plant kingdom we studied Cycas spinous, you remember, foliage leaves we call, right? Foliage leaves at autumn season generally yellow in color. It's because of most distributed xanthophyll, it's called as lutein. Very very important to remember. Carotenes we find in case of like red algae, brown algae, and xanthophylls are generally found in xanthophyceae. So these things, if you look at, these are the most important pigments. Not only carotenoids, chlorophyll is also associated with other pigments like phycobilins. In lower prokaryotic organisms or bacteria which are photosynthetic in lower organisms, also find with other pigments. In higher plants also they present in trace quantity. So let us discuss about, after carotenoids, let us discuss about other pigment is phycobilins. Okay, let us discuss about phycobilins. Right. Phycobilins generally they are proteinaceous compounds, protein compounds. As they are proteinaceous in nature, they easily get destroyed by heat. They easily will get destroyed by heat. Okay. Phycobilins are protein compounds and they easily get destroyed by heat. And these phycobilins are of three types. Okay, so they are nothing but, let us take the first one is allophycocyanin. Phycocyanin generally present in blue-green algae. Blue-green algae. Okay, this is allophycocyanin, this is first category. Then apart from this, let us compare other two. The second one is phycocyanin and third one is phycoerythrin. Phycocyanin and phycoerythrin. Allophycocyanin, it's a pigment present in blue-green algae and some members of bacteria and some of the algae. So coming to this, phycocyanin is also the pigment. It's present in more quantity, more in case of brown algae and very less in case of more pigment meaning more phycocyanin is present in brown algae and less pigment, very less phycocyanin is present in case of red algae. Phycocyanin is the major pigment and present in more quantity in brown algae and very less quantity of phycocyanin is present in red algae. Quite reverse of this phycoerythrin. Here if you take, more pigment of phycoerythrin is present in red algae, okay, and less pigment is present in case of brown algae. Okay, so you know that very clearly. Phycoerythrin in plant kingdom algae we studied, phycoerythrin is more in red algae and less quantity is present in brown algae. Phycocyanin more quantity found in brown algae and less quantity in case of red algae. Even phycobilins are also the major components. So what are the pigments we studied? Chlorophyll we studied, Then we studied carotenoids, right, second category. And what is the third category? Phycobilins. All includes what are called pigments involved in photosynthesis. Then let us talk about a nature of light. Let us talk about a nature of light. When you talk about nature of light, who is a major source? Sun is the one who acts as the source of energy. From sunlight, light energy we get it. The light energy we get it in terms of, we take that as electromagnetic radiation. So we capture energy, we take energy from electromagnetic radiation. Of course, you study more in physics and your chemistry. Let us relate it to photosynthesis. Why? Because for photosynthesis, light is the most essential pre-requirement, most key role it plays. You know that. So electromagnetic radiation, mainly we talk about visible light. Right? All of you know it very well. It starts from 923 nanometers to 763 nanometers. Generally it is referred as visible light. But before 390 nanometers, when it gets exposed, then it is UV radiation, and beyond 780 nanometers, after that you can see higher radiation. Yes. So before 390 is UV radiation. After 780 nanometers, 790 nanometers is IR radiation. In between what you can find is nothing but visible light. What is visible light? VIBGR. So it's called VIBGR: violet, indigo, blue, green, yellow, orange and red. VIBGR. Okay. So here if you take first one here, you know T.W. Engelmann. Engelmann is a scientist who studied what? A first absorption spectrum. What they studied? First absorption spectrum. How first absorption spectrum is being studied? With the help of experiment on cladophora. What is a cladophora? It is nothing but a green algae which is combined with what? Aerobic bacteria. Which is combined with aerobic bacteria. Did you remember the experiment? T.W. Engelmann is a scientist where cladophora is a green algae on which he done experiment by passing the prism of light including aerobic bacteria, and he studied and discovered a first absorption spectrum. So what you have to understand in this, different pigments absorb a different wavelength of light. Different pigments absorb a different wavelength of light. Okay, so what we can see is the ability of different pigments, I repeat, the ability of different pigments to absorb a different wavelength of light is called absorption spectrum. It's called absorption spectrum. And this absorption of light can be measured with an instrument called spectrophotometer. Spectrophotometer, very, very important. Okay, so what we have understood by paper chromatography, we could able to see different pigments. How we can see different pigments on the TLC paper? Different pigments, they absorb different wavelength of light. So the ability of different pigments to absorb different wavelength of light itself is called absorption spectrum. On spectrum you can see absorption of light by pigments, and that can be measured with instrument called spectrophotometer. Now when a graph is plotted here, so this is like something like 400 or 500 and 600, 700 like this. This is x-axis. Let us take wavelength in nanometers. Wavelength of light in nanometers. And let us take on y-axis. If you look at on y-axis. So here absorption of light by pigments. What you can see? Absorption of light by pigments, and that we measure in terms of percentage. What you can take on y-axis? Absorption of light by pigments, that you can take on y-axis, percentage. Let us take 50% of absorption of light. Then here. Then what you can see here? Now let us take different pigments, no? So let us take, this is chlorophyll A. This is chlorophyll A. What is a chlorophyll A color? This is blue color. Why this is blue color? In the blue region it shows highest rate of absorption of light. Why? Because in visible light, violet, indigo, blue. When light is passing with the spectrum, and in blue light, chlorophyll A shows maximum absorption of light. Chlorophyll A shows a maximum absorption of light. Absorption of light. That is in which region? Blue region. That is in which region? Blue region. Blue region, chlorophyll A shows maximum absorption of light. Then followed by what? Then followed by, this is chlorophyll B. This is chlorophyll B. The chlorophyll B shows a maximum absorption of light in red region. In red region. Which is red region? This is red region. So here chlorophyll A shows maximum absorption of light in blue region. That's very important. Maximum absorption of light means more light is absorbed by chlorophyll A in which region? Blue region. Then followed by chlorophyll B shows maximum absorption of light in red region. So in red region, who shows maximum absorption? Chlorophyll A. So these two are very important points. I hope you got it. Okay. First graph is for chlorophyll A. Chlorophyll A in blue region, it shows maximum absorption of light. Another pigment chlorophyll B, in it absorbs more amount of light, it takes more amount of light in red region. Then followed by and other pigments like xanthophylls. So this is called as a first absorption spectrum. This is what is called as a first absorption spectrum. Okay, so when you take first absorption spectrum, and this is followed by, actually you could able to see first action spectrum. First you could able to study first action spectrum. Why action spectrum? Why do you call this as action spectrum? Photosynthesis is measured. Photosynthesis is measured by oxygen release. So the rate of photosynthesis is measured by oxygen release. Oxygen is released where? In the green algae. So if you take the culture plate, here algae is there. Algae undergo photosynthesis and releases oxygen. What is the indication that oxygen is released? There you find the growth of more aerobic bacteria. Actually Eagleman has given first action spectrum. First he has given action spectrum. Later he understood that, oh, it is because of absorption of light. Then later what come is called as absorption spectrum. Of course absorption spectrum and action spectrum also they are related to, they are related to Emerson scientist. First absorption spectrum, first action spectrum, both of them are more closely related with the experiment done by Emerson. Both of them mostly they come under the credit of Emerson. Emerson has given the study of these two spectrums given as it's called as Emerson enhancement effect. Emerson enhancement effect. So we will discuss about Emerson experiment. We will come back to this part later in continuation of the class. Okay. So mainly Eagleman is more closely related to the action spectrum and absorption spectrum. Some extent he also contributed to the absorption of light and mostly contributed to the action spectrum. Okay. But according to the early experiments, introduction about the scientist and discoveries, they already we discussed about T.W. Eagleman. You follow the statement very clearly, okay, then you will get the clarity. But some extent is related to the other spectrum also, where a rate of action spectrum meaning, first action spectrum meaning rate of photosynthesis is measured with the oxygen release. This is called as action spectrum. Absorption spectrum means this is absorption spectrum. How pigments are absorbing the light, it is called as absorption spectrum. How pigments absorb different light is called absorption spectrum. In absorption spectrum, chlorophyll shows high absorption in blue region, chlorophyll B shows high absorption in red region. That very important. Then coming to action spectrum. Now let us talk about action spectrum. In action spectrum, what we are doing? We are measuring rate of photosynthesis, how much photosynthesis is taking place, that can be measured with amount of oxygen released in the process. More oxygen is released, more rate of photosynthesis. Less oxygen is released, less is the rate of photosynthesis. So even this is being plotted with the graph, same like this. Some here we can take wavelength of light absorbed. So here we write wavelength of light, that is measured in terms of nanometers, and in this rate of photosynthesis by oxygen release, how it is measured, how much photosynthesis taken, how did we come to know? By measuring the oxygen release. Okay. So here this is on y axis, this is on x axis. When a graph is performed here, the graph also showed more oxygen released in blue region and then followed by red region. That means here what is released? More oxygen is released. More oxygen means what? High rate of photosynthesis. High rate of photosynthesis. That happened in which region? Blue region. That happened in blue region. More oxygen is released, meaning this is the region, more rate of photosynthesis has taken place, and that is in which region? Blue region. Then followed by, so little high you draw because chlorophyll B also does. So here if you take, then coming to red region, in red region also, so this is red region. In red region also, here also it is recorded as high photosynthesis. It is recorded as high photosynthesis. So in terms of these two regions are called as photosynthetic regions, and this is also called photosynthetic region. I hope you are getting. Here more rate of oxygen release, it is because of high rate of photosynthesis that happened in blue region, and this region is called photosynthetic region. Obviously in red region also there is high rate of photosynthesis, and this is also called photosynthetic region. That means here if you look at in terms of photosynthesis, which one is the highest in photosynthesis? Very important note point I am giving you. Photosynthesis in terms of this, first is blue light, then followed by red light, then followed by green light. Better I will write separately, very important. So here you can take, very important, when we say high rate of photosynthesis, or we can talk high absorption of light, high absorption of light. Okay, high rate of photosynthesis, high absorption of light. Remember, the first comes is blue light. This happens in blue light region, then followed by red light, then followed by green light. Okay, so high rate of photosynthesis means this is action spectrum. Okay, rate of photosynthesis means action spectrum, absorption of light means absorption spectrum. When both are compared, then maximum occurs in blue light, then followed by red light, then followed by green light. But whenever the question is given in terms of high transmittance of light, then comes green light, followed by red light, followed by blue light. What is meaning by high transmittance of light? Green light is more absorbed, it will get more transmitted into the parts of the plant. As more green light is getting transmitted, that is why the parts of the plant look green in color. That means absorption of green light, transmittance means it goes and reflects. More transmittance means it will get transferred into the parts of the plant. More it will get transferred into the parts of the plant. Chlorophyll is absorbed more with the green light and less is the reflection. So, parts of the plant looks green in color because green light shows high transmittance of light. Now, let us compare with action spectrum and absorption spectrum, that is rate of photosynthesis and absorption of light, or first we have to say absorption of light, then followed by rate of photosynthesis. Why? Because after absorbing light, then only photosynthesis takes place. So, both here we are going to draw both same like 400, 500 or 600. What we are taking x axis is same, that is wavelength in nanometers. So, we are just taking comparison between absorption spectrum and action spectrum. Absorption spectrum and action spectrum. Okay. So, we are taking both here absorption and oxygen release. Okay, that we talk in terms of photosynthesis. That means what are the things here? We are taking pigments and what is their absorption rate and how much photosynthesis they have done. Pigments, how much they absorb the light, absorption of light by pigments, and when they undergo photosynthesis, how much oxygen is being released. Both we are taking together. That means we are taking both absorption spectrum graph and using that we are comparing with the action spectrum. Then if you look at, so when you talk first action, that is or you take absorption, better you can write. It is like this. Action spectrum I will draw first. Action spectrum. This is action spectrum. and absorption spectra also somewhat like this. Okay, so this dotted line says this is absorption of light by pigments. That means it speaks about what? It speaks about absorption spectrum. Okay, and very thick dot line says, this says release of rate of photosynthesis by release of oxygen, and that comes under action spectrum. Okay, so what we are comparing? So here generally what is the final conclusion out of this? The dotted line says that how much light is being absorbed. Light we are measuring in terms of wavelength, that is in nanometer. The more is the sunlight absorbed. Where more is the sunlight absorbed? By chlorophyll A. In which region? Blue region. Right? Then they undergo high rate of photosynthesis. That means here absorption spectrum, action spectrum. This is absorption of light. Dotted line says absorption of light. More is the light is absorbed by the pigment, then more is the rate of photosynthesis. Why? Because there is more oxygen released. So this is action spectrum. This is when dotted line absorbed is more, that is by chlorophyll A. In blue region automatically there is a high photosynthesis, then more oxygen is released. Okay, this is oxygen release. Yeah, this is oxygen release. More oxygen is released. So this is action spectrum and this is absorption spectrum. More light is absorbed, more oxygen is released because high rate of photosynthesis. Then it falls down with the chlorophyll B, xanthophylls, carotenoids. Then coming to chlorophyll B here. So this is by chlorophyll B. Which region is this? This is nothing but red region. Again in red region from 600 to 680 nanometers. I will let you know later. What is VIBGR? VIBGR you know it very well. So this is violet, indigo, blue, green, yellow, orange and red. So which are the regions you show with high rate of photosynthesis? Blue region. So when light is passing, indigo and violet indigo, when it comes to blue light, when it comes to blue region, chlorophyll A is showing high rate of photosynthesis with the more release of oxygen, Then remaining pigments, remaining green, yellow, orange, light is falling down. Coming to red region, red region means 600 to 680 nanometers, that I will give you later, because here we have a concept called red drop. Emerson enhancement effect. So this we will study under Emerson effect, that we will discuss for next class. So red region, regarding red region we have to study Emerson's effect, because Emerson is a scientist who studied about this. In red region chlorophyll B shows maximum absorption of light and more photosynthesis. When it is more photosynthesis, again there is a release of more oxygen. That means here we say blue light and red light, means more is the absorption of light is by dotted line. Dotted line says absorption of light by pigment, that is absorption spectrum. Obviously when more light is observed, then this dark line says this is action spectrum, that this is a place where you can see more release of oxygen. Automatically it is understood that there is a more rate of photosynthesis. Now what is you can tell me now here, that is photosynthetic regions, photosynthetic spectrum or photosynthetic regions if you take. What are the two lights we have to take? One is a blue light and other one is a red light. Blue light as well as what we find is a red light. So, this is the comparison between absorption spectrum and action spectrum. But what is important here in this, nothing but this is very important. This part is very important. Why that part is very important? When you take that particular part, in this case what you can see is whenever the question is in terms of high absorption of light by different pigments, you have to understand the question is speaking about absorption spectrum, okay. And when the question is speaking in terms of oxygen release and the graph plotted between wavelength of light and rate of photosynthesis, that is measurement of production of oxygen by rate of photosynthesis, that speaks about action spectrum. This part is very important. That means what you have to understand, whenever the question is high rate of absorption of light or high rate of photosynthesis, what is the order of lights you have to remember? First goes to blue light, order is then followed by red light, then followed by green light. Suppose if the question is about high transmittance of light, high transmittance of light means the more green light penetrates into the deeper parts of the plant, less is the reflection. More is the absorption, less is the reflection, more it is getting transmitted, green light more it is getting transmitted into the parts of the plant. That is why all parts of the plant they look green in color. So, when you talk in terms of high transmittance of light, green light, listen the question carefully. When you talk in terms of high absorption of light, high rate of photosynthesis, the order of light is blue light greater than red light greater than green light. Suppose if the question is high transmittance of light is first comes green light, because green light shows high transmittance, all the parts, sometimes you will get a general question, all the parts of the plant are generally green in color because of what? Because of green light, because it shows high transmittance. Remember this and feed it in your mind, this is very important point. Suppose the question is given in terms of high transmittance of light means then first comes green light, not to confuse, not the blue light, first comes is green light is greater than red light which is greater than blue light. So, green light high transmittance, compared this less transmittance for red light, compared to red light less transmittance is for blue light. So, these things you have to remember. So, in this box you have two questions, high maybe absorption of light, rate of photosynthesis, this is the order. If the question is in terms of high transmittance, you have to go from down to up, that is green, red, blue. I hope you understood. Now, I will just quickly revise with this comparison between absorption spectrum and action spectrum when you take. So, here the dotted line what you can see is absorption of light by pigment, that speaks about absorption spectrum, and the dark line which shows here, that is rate of photosynthesis by release of oxygen, that speaks about action spectrum. As usual we take two columns, x axis and y axis. Now, x axis in all the graphs we are taking wavelength in nanometers, that is light wavelength, different wavelength of light. On y axis we are just comparing both absorption of light by pigments and also we are comparing how much oxygen is produced in photosynthesis. Both we are taking the parameters under this. Two graphs are compared here. Then when dotted line, if you see the dotted line here shows maximum absorption of light in blue region, same like earlier we discussed too. I am just keep repeating, why? Because the questions whatever you got on absorption and action spectrum, you are not supposed to get stuck up with the questions, that is why again I am repeating. Hopefully you can follow this again and again. I am repeating because this you need to focus, again listen carefully. I do not want you to lose the question if at all it is given on absorption spectrum and action spectrum, that is the reason. Same story here again in comparison. So, if you look at dotted line absorption of light, so that is more absorption of light by chlorophyll A, that happens in blue region and that comes under absorption. Then followed by action means what action is happening means in the same blue region more rate of photosynthesis that is measured, you can see more oxygen is produced by green algae, aerobic bacteria accumulated over there. So this is action spectrum, this is absorption spectrum. So they understood that chlorophyll A in blue light shows high rate of photosynthesis with more release of oxygen, that is it. Then with chlorophyll B, C and D, E, then followed by carotenes, xanthophylls, it is coming down. Then again chlorophyll B is rising up, especially in red region, it is rising up. Why? Because here also again more absorption of light, that is dotted line, followed by more release of oxygen, that is dark line. So in chlorophyll B in red region, it also shows high rate of photosynthesis with the more release of oxygen. So this happens, red region is from, as I told you, 600 to 680 nanometers, but remember after 680 nanometers, so I am showing here after 680 nanometers there is a decrease in photosynthesis. After 680 nanometers there is a decrease in photosynthesis, that is studied by Emerson and it is called Emerson effect, that we call red drop, that we will discuss. It is also called as red drop. Why do you call this as red drop? Because after 680 nanometers the rate of photosynthesis falls down, drops down, that is why it is also called red drop, and this is explained by Emerson effect, that we will discuss in the next class. So photosynthetic spectrum, if you take mainly blue light and red light. Okay, so this is nothing but comparison of action spectrum and absorption spectrum.